Hi, my dear students. We're going to continue and finalize our discussion on cell injury. And so our last topic, or subtopic, I should say, on cell injury is cell death, which is obviously an irreversible form of cell injury. So in previous presentations, I've already mentioned the two major forms of cell death, which are apoptosis, which literally means, if we translate it, the dropping, let me use a different color, the dropping of leaves. Apo referring to leaf. So literally cell dropping, essentially. And then there's also necrosis. Autophagy is also, just like apoptosis, a form of programmed cell death. And I briefly mentioned it in our discussion on cell injury when lysozymes are damaged to where they leach or leak out their enzymes to where organelles on the inside of the cell are being digested. We will first focus on apoptosis before we get to necrosis. And as we discuss these two, eventually necrosis, it's going to be really important for you to be able to differentiate between these two forms of cell death. They may at first seem or sound similar to you, but there are very distinct differences. Um, Apoptosis is a very controlled and a very keeping things in line type of cell death. So when it occurs, it's not going to create this cascade effect of lots of cell death in a tissue, which is different in the case of necrosis. Apoptosis is often called cell suicide. And it happens to just get rid of cells that really just don't belong anymore. Either the cells are damaged due to overuse, or it could be that they are just reaching their old age. Yes, cells age as well, and we'll talk more about how cells actually show their age. So apoptosis, you already have a bit of a feel with what regulates it because we've discussed it before. Remember how calcium overload can trigger apoptosis in cells. So the levels of intracellular calcium are going to dictate whether or not apoptosis might be occurring. But there are also hormones, and then we're going to talk about two types of genes that we will talk a lot about when we talk about the one of our topics in a future block, and that is oncogenes and tumor suppressor genes. But believe it or not, they play a role in cell death, namely apoptosis as well. So as I already said, apoptosis occurs to get rid of cells that really don't belong anymore. And it's a very important process. So if there is some kind of a um, mechanism in the body that prevents apoptosis from occurring on a regular basis, we might be looking at cancer. Um, and therefore, one of the primary mechanisms of cancer therapy is to actually go after those cells that are not committing apoptosis. So when cells have been mutated enough to where they do not go through apoptosis anymore, they could start causing cancer. So keep that in mind. So again, apoptosis is a normal process. And you go, what do you mean? As mentioned, cells have only a certain length of life and then they need to die. And literally, there's alterations in their chromosomes, not mutations, but there are going to be changes to what we call their telomeres that indicate that they have reached a senescent stage and it's time for them to go. But also, as we develop as a fetus, all kinds of um, cells go through the process of apoptosis. So all of you have seen images of very early, well, I shouldn't say very early, but fetuses that still have a little bit of webbing between the fingers, not so apparent for you to see in the toes, but in the little fingers or between the little fingers, and that webbing needs to disappear, and that happens because cells go through apoptosis. Your skin cells are constantly flaking off as you're constantly losing hair. All of these cells and the cells of your skin and your hair are called keratinocytes, go through apoptosis. When we develop as a fetus, we produce, or we develop all of these neurons, so many, and most of those are going to go through apoptosis. I've got to be careful, maybe not most of those, but many. And a lot of that has to do with the fact that neurons need to learn whom to synapse with and make a lot of mistakes. And when they make mistakes, they go through apoptosis. Immune cells go through apoptosis all the time when they go through their process of self-tolerance, or when cells go through phagocytosis, often after they've committed the act of phagocytosis, they go through apoptosis. And then finally, we lose, or we overturn, I should say, the lining of our digestive tract. Remember, that is simple columnar epithelium about once a week, give or take a day. It probably depends on the person. And so those cells go through apoptosis as well. And of course, that means that there are stem cells to replace the epithelial cells that we lose both in the skin and the GI tract. So apoptosis is a very normal process, and it needs to happen to prevent cancer. So it's interesting when cells become injured. Let's say if we look at a heart attack, right? A fancy way of referring to it as a myocardial infarction, the majority of the cells actually die by suicide, by apoptosis, and only about a fifth of them are going to die by necrosis and then create all kinds of inflammation, as we will see in a moment. Hormones can also trigger apoptosis, right? Every month or every 28 days, give or take a few days, girls go through a period, or they have their menses. Even when a woman goes through menopause or pregnancy, we're going to see that there's a lot of apoptosis occurring. And then apoptosis is also going to occur not as a result of healthy, normal stimuli, but also due to pathological stimuli. So pathogens, environmental toxins, etc. Medications, if they're overused, can also trigger apoptosis. So it's going to be really important for you to be able to distinguish between the mechanism of apoptosis and the mechanism of necrosis, because the consequences of these two forms of cell death are also very different. So in apoptosis, we're not going to see that the cells swell and then spill their contents. Not at all. They do almost quite the opposite. They're going to shrink and excuse me, they're going to use a very minimal amount of ATP as well, by the way. So yes, for apoptosis, we are going to depend on a small supply of ATP. Because the cells are not going to swell and burst, they're not going to spill their contents into the interstitial fluid, so no spilling, and therefore there will be no inflammation. You can already see where this is going clearly in necrosis. It's just going to be the opposite. Apoptosis is one of these processes. You'll see it depends on a cascade event, or cascade events, I should say. Therefore, once it starts, it cannot stop. So it's one of these things that needs to finish once it gets going. And there are two pathways that we will take a look at: the intrinsic or the extrinsic pathway that can cause apoptosis. So the intrinsic apoptosis pathway, and anytime the term intrinsic is used, it means that it's from within a cell or from within a structure that we're discussing. So the triggers are not coming from some external source. So the intrinsic apoptosis pathways is triggered by intracellular signals. It could be that the cells are indicating that they have grown old and need to die. So cell senescence, there might be an accumulation of a bunch of ROS. We don't have enough ATP production. There might be calcium overload. And all of these things, well, except for cell senescence, you're pretty familiar with. And then we're also going to talk about a protein called P53, which we will talk a lot about in cancer as well. And this protein is the product of a gene that codes for P53, and that gene is called a tumor suppressor gene. So the protein is P53, and the gene is a tumor suppressor gene. We have lots of tumor suppressor genes in the body, but this one is the best understood. So tumor suppressor genes, and therefore their proteins, are pretty amazing. P53 is the most well-known tumor suppressor protein, and it plays a role in the cell cycle. So here we're looking at the intrinsic pathway where P53 can or has control over whether or not the cell cycle continues or apoptosis occurs. So let's take a look at exactly what is going on here. So let's say that a cell has incurred cell damage. Let's say that hypoxia has caused the DNA damage, right? So some bases have been damaged, perhaps a whole gene has been damaged. Somehow this is going to trigger P53 to become increased in the cell, and it can force the cell cycle to be arrested. So P53 is going to force the cell cycle to be arrested such that the DNA can be checked. There's various mechanisms and proteins that check the DNA. So the DNA is checked, and if possible, it's repaired. And if the DNA can repair itself, then P53 will make sure that the cell cycle continues. And therefore, mitotic divisions can continue at a normal controlled pace. If, on the other hand, the DNA cannot be fixed, so the DNA repair cannot occur, we're going to instead see that P53 will trigger apoptosis and therefore eliminate the damaged cells. So both of these sides here, whether the cell cycle restarts or we eliminate damaged cells, is going to make sure that we maintain cellular stability, but also we maintain genetic stability, because remember, these cells had damaged DNA. So this is how P53 functions in what we call the intrinsic pathway. Let's now take a look at how P53 functions in the extrinsic pathway. So the extrinsic apoptosis pathway is, interestingly enough, often referred to as the death receptor dependent pathway, while the intrinsic one might be called the death receptor independent pathway. And indeed, we're dealing here with a receptor called FASR. By the way, often when you see an abbreviation like this followed by a capital R, the capital R is implying a receptor. And it binds molecules, it binds ligands that function as death factors. And the obvious ligand would be a Fas ligand, Fas L, with the L standing for ligand. But there's also another one called tumor necrosis factor. And those are just a couple of examples. And we'll look at a diagram in just a moment to make this a little clearer. So when these chemicals which can bind with our receptor, so these are ligands that can bind to our receptor, when they bind and therefore act as death factors binding with our death receptor, they're going to be able to trigger apoptosis. By the way, the kinds of cells that secrete these kinds of death factors are macrophages especially, but also the two cells that go after cancer cells and damaged cells, your T cytotoxic cells and your natural killer cells. Remember, these two their mechanisms of killing are very similar. So here we see two diagrams representing the extrinsic apoptosis pathway. On this side, we see our cell membrane. Here we see our famous death receptor. And here we have our ligand, which is Fas-L. So this is our Fas receptor to which our ligand Fas-L binds. While over here, similar thing, except that our death receptor this time is binding tumor necrosis factor. And notice that this abbreviation is, there's a bit of a typo. This should say TFN. I'm sorry, tumor necrosis factor. I just copied the wrong thing. TNF for tumor necrosis factor. Okay, so that's the setup here. And what happens next? What is it that really leads to apoptosis? Well, we're not going to, of course, study the detailed biochemistry here, but what we're ultimately going to see after a death factor binds to the death receptor is that a series of events occurs. It's kind of like a cascade event. And we only see a couple of steps here, but there are many more. And ultimately, it leads to enzymes called caspases. So caspases are activated. It doesn't matter whether it's what the ligand is. So the binding of a death factor to the death receptor triggers these caspase enzymes, which then leads to a series of events that eventually cause apoptosis. So from now on, when I show you diagrams that sometimes look very complex, and we will not always look at all the details of biochemical pathways. That's well beyond the focus of this course. But look for these guys, these caspase enzymes, and always associate caspase enzymes with apoptosis. So caspase enzymes, apoptosis. If you can do that, you're already a huge step ahead of everybody. Now, about these caspase enzymes, they're also formed in the intrinsic pathway. So the diagram I showed you for the intrinsic pathway was extremely simplified, but you need to be aware of the fact that both pathways, both of them, are going to end up producing caspase enzymes. So really what happens in apoptosis, as I mentioned before, we're going to see the shrinkage of the whole cell. So not only the whole cell, but even the nucleus, the cell membrane begins to shrink. But what is important to note here, so here we have our normal cell, and then it's going to start shrinking. What is really important here is that the cell is not going to fall apart. So it'll just sort of shrivel up, and it forms what they then call blebs. That's what these are called. And they can eventually pinch off. So the cell membrane is not going to lose its integrity. This is really important for you to get here. The cell membrane remains intact, but instead it pinches off. And literally, by it pinching off, it keeps the contents of the cell in these little bodies that we call apoptotic bodies, little bodies as a result of apoptosis. So apoptotic bodies. And now these are small enough to where a macrophage or some other phagocyte can say, oh yeah, small enough, I can go after this and phagocytose this. And so this is a very cleanup. It's not messy at all. It's very controlled. Um, this is basically, a maid service a professional maid service that is extremely organized, where everything is put back in place by the time they're done, and everything looks spotless. Let's put it that way. Just a quick little reminder that a form of cell suicide is also autophagy. Remember that autophagy is literally meaning self-digestion or self-eating, auto, and then phagy referring to eating. And cells do this often in an attempt to get rid of organelles and proteins that are damaged or old. and they can then recycle the components, the breakdown products, and make new ones with them. So we've looked at this before, particularly when we talked about lysosomes and proteasomes. So we just finished discussing apoptosis. So then how's that different from necrosis, which is also cell death? Well, necrosis, let's just say it's messy. It means that you have hired a maid service uh, from some ad written on a cardboard out there on the street, and by the time this person, because it's one person, arrives, you realize this is a young kid that doesn't know what the heck he or she is doing, and you end up with a bigger mess than what you started out with, basically. It's not really the best analogy, but it might trigger your memory a little bit better when you take your tests, um, and remember that apoptosis is really tightly controlled and not messy, and there's no inflammation, nothing leaks out. Necrosis is a different story. It's so messy that it's going to interfere with the functioning of the cells surrounding where necrosis is occurring. It's going to interfere with replication of cells, with tissue regeneration, just to name a few. So in necrosis, we do have swelling and we do have damage to the cell membrane to where it doesn't maintain uh, its continuity. So there's going to be interruptions of the cell membrane. It's going to break apart. And next thing you know, all these cell contents are going to end up in the interstitial fluid, and that's going to trigger an inflammation. We haven't studied inflammation yet. We will, and you will see what, inflammation can be a good thing, but it can also get out of hand, and often it can lead to damage to surrounding cells. The other big difference is that necrosis does not need ATP. Apoptosis needs a small or low level of ATP, so it needs a little bit of energy to get cranking, but necrosis does not. So the mechanism of necrosis is quite different from apoptosis. So what we're seeing happening is that for some reason these enzymes begin to digest the cell from within. These enzymes are very unregulated, and consequently we're going to see that um, calcium levels on the inside of the cell begin to increase, not just because um, little by little we're going to see that the cell membrane is damaged, but also because organelles such as smooth endoplasmic reticulum and mitochondria that store calcium are being damaged, right? They're being digested. But as calcium levels begin to rise inside of the cell, we're also activating a variety of enzymes, such as your phospholipases, proteases. We need those because, I'm sorry, we don't want those to touch our cell membrane, because that means that we're digesting or taking apart phospholipids and proteins that make up our cell membrane. So we see even more problems with our cell membrane. Next thing you know, all these contents that are being taken apart inside of the cell, including the cell membrane, begin to just spill over into the interstitial fluid all over the place. Big flood, big soup of all of these intracellular things, and that is going to attract immune cells, and next thing you know, you have this big inflammatory response. And anytime you hear the word inflammation, particularly if it's an intense response, you can automatically assume that there's going to be damage to neighboring cells and possibly the whole tissue. So I've given you here a very nice overview of apoptosis and necrosis. And here's a continuation of that table. There's one thing I didn't really talk at all about when I discussed apoptosis and necrosis, and that is the nuclear DNA. What we see in apoptosis is that the way the DNA is dealt with is by very controlled and I guess I use the word programmed here, splitting up of the DNA into little pieces and then it becomes part of those apoptotic bodies. Remember those little blebs that pinch off? That is not at all the case in necrosis. In necrosis, the DNA is just broken up into random pieces, and they become part of that soup that leaks out and spills over into the interstitial fluid. So as I said, necrosis can create widespread damage. And so here we're going to take a look at some forms of necrosis. I need to prepare you. Some of these images are not pleasant to look at, but this is how it goes when necrosis hits the body. So we need to face this. So in these different forms of necrosis can sometimes be hard to memorize. So you're going to have to find ways to, you may want to make some tables to help you keep them separate. There's some key words and key terminology that you can put in these tables to help you separate or distinguish between different forms of necrosis. So in coagulative necrosis, listen to the name coagulative. So remember, coagulation is a different way of how we refer to blood clotting. So coagulation doesn't always have to mean the coagulation of blood. You can have coagulation of proteins, for instance, too. But essentially, what results from coagulation is that something more solid is formed, and that is the case here in coagulative necrosis. You form a solid mass of damaged cells, and the reason for this is because the damaged cells are not being taken apart by proteolytic enzymes. And why is that? Well, in this form of necrosis, often the injury leads to acidosis, meaning that acids are formed, which then, as you know, when the pH drops, destroy a variety of proteins, but especially what impacts the formation of coagulative necrosis is the denaturation of structural proteins, meaning, again, part of the cytoskeleton and lysosomal enzymes that that normally take apart organelles and cell components. So these proteins and these enzymes, which of course are also proteins, stay intact, and therefore they literally cannot take apart the damaged cells, and we end up with this solid mass. This is the most common form of necrosis. So coagulative necrosis is the most common form, and because it's a solid mass, it takes a few days for it to dissolve, so it takes time. Some other characteristics of coagulative necrosis is the fact that you can really see a demarcation line um, between the still healthy vascularized tissue and the necrotic tissue. So I'm going to attempt to illustrate this to you. Let me grab another color here. I'll use blue. So notice this creamish color here, right, on the inside of this adrenal gland versus the then more reddish colored vascularized healthier tissue. So this is really characteristic for coagulative necrosis. And also, this is going to be a dry form of necrosis. So now you have some keywords to throw into that table that you're going to be creating for yourself to help you distinguish between the different forms of necrosis. Now, what we haven't discussed yet is what tends to cause this form of necrosis, coagulative necrosis, and that's primarily ischemia, meaning uh, poor blood flow, poor perfusion, as would happen when a clot blocks the flow of blood in the heart, for instance. So it's not going to be due to any kind of severe, nobody in a car accident where your body is slammed into the steering wheel, or toxins, or any kind of an immune response. It's really due to the fact that there's that somehow the blood can't flow properly. So our next form of necrosis is called liquefactive necrosis, and as the name says, liquefactive, it is wet. You can see that very well here on this picture. And the area of necrosis is really a space that's filled with a fluid, particularly after it becomes the area becomes dissolved by white blood cells. Um, and so it can be a cyst type of an area or an abscess type of an area. We see this often in the brain. What happens here in the case of liquefactive necrosis is that the cells that are injured die, but their enzymes are again not destroyed. In addition to that, often we see that the white blood cells that came towards the injury site in response to start the inflammatory process, they tend to remain behind and create this pus, creating also the very liquid type of necrosis. Liquefactive necrosis can also be caused by ischemic hypoxia. So we saw that coagulative necrosis is also caused that way. But in addition to that, it can also be caused by an infection that results in inflammation and attracted a bunch of white blood cells. And these white blood cells contain many enzymes that then contribute to that very soft center of the cyst or the abscess and creates that very liquid look. As mentioned earlier, this is a form of necrosis that is very often seen in brain tissue. There's also a form called caseous necrosis, and you know, in Spanish, queso means cheese. In my language, gas means cheese. So maybe that'll help you remember caseous necrosis has that cheese-like look. So it's a type of necrosis, as you can see here, that almost like cottage cheese kind of. It's very fragile and it kind of breaks off. It's soft, white, cream. And this is very characteristic in lungs that lungs with tuberculosis, meaning they're infected by your famous Mycobacterium tuberculosis bacterium. And what we see happening here in this form of necrosis is literally the cells that are necrotic and dying or dead have been put in these little isolated pockets that we call tubercules. They're literally isolated from the healthy tissues. We will come back to discussing these tubercules and how dead cells are walled off in these areas in another topic down the road. Fat necrosis, as the name says, is necrosis of fat tissue or adipose tissue, and it has this creamish to whitish color, maybe light gray. And we see this often in adipose tissue of the breast. You know, breasts are very rich in adipose tissue. So is the subcutaneous layer underneath the skin, or you can call it the hypodermis. We even see it in pancreatitis. And let me first explain what happens in fat necrosis, and then the problem with pancreatitis starts to make more sense. So what happens in fat necrosis is that enzymes, they break down triglycerides. And as they break down the triglycerides, remember triglycerides are made up of glycerol and fatty acids. These fatty acids are then going to be able to bind free-floating calcium ions, and we call this process saponification, or we form calcium salts with fatty acids, and they precipitate as calcium soaps, as it's sometimes referred to. So this process of saponification we see happening when a pancreas is inflamed, such that the pancreas produces lots and lots of enzymes. The pancreas produces enzymes that break down all major biomolecules, right? All four of them, including secreting lipases, which can break down triglycerides. So now we see that Lipases are dumped into the abdominal cavity because the pancreas is damaged, it's inflamed, and the lipases are not staying where they're supposed to stay. And consequently, we're going to see that surrounding our pancreas, we're going to see that adipose tissue becomes saponified. I guess that's a verb I'm allowed to use. And finally, we get to gangrenous necrosis or gangrene. And we're going to see that there's actually different forms of gangrene. So we're not quite done yet, but we're getting there. Gangrenous necrosis really just implies a very large area of necrosis, and it's a combination of the two first forms of necrosis that we discussed, the very solid form and the very liquid form. So what's happened here is that the ischemia is responsible for causing the tissue death, but then the bacteria that build up and the white blood cells that are attracted to the dead tissue cause the liquefactive type of necrosis. In gangrene, we're always going to have a horrible smell, and it's going to look quite black. So there are three types. Let's take a look at them. Dry gangrene, as the name says, is a very dry form of gangrene. We see the black tissue here, obviously, and it's very dry looking, but also notice that there's a very clear line separating the healthy tissue from the necrotic gangrenous tissue. And this is because the blood supply that is going into these toes, so the arterial supply, is somehow blocked or partially blocked. So we have a form of ischemia going on. And this is very common in the limbs, and as you see here in the toes. Unlike wet gangrene, where we do not have a clear demarcation line. And unfortunately, I couldn't find a really good picture to illustrate wet gangrene. But this time it is due to the fact that there's a blockage in the venous drainage. So, excuse me, I'm just going to, for the heck of it, use the toes again. Maybe not the best example, but here is my foot. So this time it is and I'm going to use blue, it is the draining that is blocked, the draining of the blood. So these are representing veins. So the blood is not draining the tissue very well. And consequently, we do not have that demarcation line. And the blood collects here, creating a more wet type of gangrene. Not only is it moist and black and tender, but it's going to be cold and swollen and pulseless, and little blebs will often form as well. The pulseless, again, due to the fact that there's issues with the venous drainage. This type of gangrene spreads fast, and as it does, it can also dump a whole bunch of toxins in our blood, which means it is death threatening. This is a type of gangrene that needs to be watched very, very carefully. And finally, we have gaseous gangrene. As you can imagine, this is due to the formation of gas bubbles. And what else than an anaerobic bacterium that would be responsible for this? So there is a bacterium called Clostridium. You might be familiar with it from microbiology, and it can create these hydrogen sulfide gas bubbles, particularly in the muscles. You can see them here. And this is very often as a result of major trauma, compound fractures. You know, this looks like a person who might have been in a bad car accident, might have been perhaps um, squeezed in between very heavy objects, something along those lines. And this is the type of gangrene that more than likely is going to lead to amputation. So this almost wraps up our discussion of apoptosis and necrosis. Really, we're done. But there's still something fun I'd like to share with you, which is related to COVID and what we've just discussed. So let's take a look. So for about 30 seconds maximum, we're going to talk about pigs, apoptosis, and COVID. So pigs also get COVID. They are also infected by our infamous virus called the SARS-CoV-2 virus. But interestingly enough, unlike us, who when we're infected with this particular virus, we can get extremely sick and die. Pigs do not really get sick, if at all, and they also do not pass on the virus very easily at all. And this is because, unlike us humans, where we mostly depend on necrosis in order to get rid of the cells that have been infected by the virus. And of course, that leads to inflammation. And in us humans, often it leads to a very, very nasty, abnormally high, or better called hyperimmune response. Some of you might have heard of the cytokine storm in people who suffer from severe COVID. Well, pigs don't do that because the cells in the respiratory tract um, of pigs, they mostly go through apoptosis, while we humans mostly depend on necrosis. Necrosis triggers inflammation and then a possible hyperimmune response. Apoptosis does not trigger inflammation, and so therefore there isn't any danger of some major nasty immune response. So I came across this study and I thought this was very interesting, and I'm sharing this with you. And yes, I may test you on this. That's the end of cell injury, finally.